Hello friends, welcome back to my channel Xgene and proteinX. In this tutorial video, I'll discuss about the overview of the SARS-CoV-2 spike protein. So as you know that the spike protein is the crucial protein of the SARS-CoV-2 that interact with the S2. So afterward, it get entry into the host cells. So here is the right side, that is the schematic diagram of the SARS-CoV-2, and in the membrane it attaches the triangular shape, the red color, that is the spike protein, and in the left side that figures it representing the translated region of the spike protein as well as the crystal structure and other some other structure of this protein. So here, as you see in the figure, that this is the 5' part and this is the 3' part, and here is the translated spike protein. And in the spike protein, post-translationally cleaved by the mammalian furin protein into two subunits, S1 and S2. So this is the furin that locates in the mammalian cells. So when the spike protein is produced after translation, so the furin protein, it cleaves this spike protein into two subunits, S1 subunit and S2 subunit. So S1 subunit, it largely consists of amino terminal domain, that is the NTD, so N-terminal domain, and also the receptor binding domain, so that is representing the RBD. And receptor binding domain is the important function of this protein to interact with the S2 of the host cells. And the S2 content, it includes some trimeric core. So trimeric core of this protein, and that is responsible for the fusion of this spike protein into the cell membrane. So in this figure, like here, so if it is like the spike protein and then this is the human cell, if you see, this is the human cell membrane, and here is the spike protein. Here is the spike protein. So here the S2 subunit, it responsible for to fusion of the spike protein in the cell membrane. So it attaches here in the cell membrane. So this is the fusion, this the main function of this S2. Also the fusion, this fusion here also you can see in this figure that the fusion of this protein when it is in the RBD part, and in the fusion this is the the trimeric core of this protein. So here you see the N-terminal domain, it contains the trimeric core of this protein that is responsible mainly for the membrane fusion. Here also this is the crystal structure of this protein. This is the monomeric and this is the trimeric figures of this crystal structure. So later on I will discuss also in details about this kind of crystal structure and these figures. So let's move on to the next slide. So here you see this is the trimeric spike protein, and this trimeric core is the S2 part that as I mentioned before, that it cleaved into S1 and S2. So in the S2 content this trimeric core, so this is the trimeric core, and here there are some important and some active amino acid residues that are shown when zoom in figures. So here you see there are some around 5 or 7, 1 2 3 4 5 6 7. These 7 amino acid residues are responsible for to fusion of the cell membrane. So this is the main function of these S2 proteins. So let's talk about something, the function of the different part of this spike protein. So here you see the S1 and S2. So S1, its main function of this S1 is to initiate the host cell entry. So recognition and the viral attachment, this is the S cell entry. So S1 part contain the N-terminal domain and the receptor binding domain. So receptor binding domain, this is the main and important domain of this domain. This is for the recognizing and strongly bind to the human S2 receptors. So this is the main function of these RBD proteins. And also here the S2 protein, here is the S2 subunit. So S2 subunit content, there are some this kind of part it consists of, and these are responsible for this is the fusion of this virus. FP means the fusion of this virus, and it target the cell membrane. So when the S2 target the cell membrane, then the S1 subunit contain RBD, it recognize the S2 receptor and it interact with the S2 receptor, and then it get entry into the cell into the host cells. This is how it works. So let's talk about something, this crystal structure of these proteins. So here, as you see in the figure, in the crystal structure, this is the receptor binding domain, and this is the N-terminal domain. And here its subunits are, there are two subunits, S1 and S2, and these are the fusion proteins. So as I mentioned earlier, that furin protein, it cleaves from here the S1 and S2 subunits. So this cleavage is very much important for the spike protein to work it properly. So the spike protein, it transiently undergoes conformational change between a closed conformation and a open conformation. So here in the closed conformation and the open conformation, in between the hinge like movement. So this is the hinge like movement, and it raises the RBD. So in precisely, if I say that, so that there are some conformation changes occurs in the spike protein, that is the closed conformation and the open conformation. So in between closed and open conformation, so if it is like open conformation and if it is like closed conformation, so in between there are some movement and hinge like movement raises, and in this part the RBD raises. So here is the RBD, it forms. So later on, this RBD are the main part of this protein. So this is okay, this is this part, and here is the main part of this, how this spike protein it interact with the S2. So there are some amino acid residues that are representing here. So this green color, sorry, this golden color, these are representing the S2 proteins. And also here there are some active amino acid sites are it's already marked here, and the blue color it's showing mainly the spike protein RBD, because here it's showing the RBD only, because RBD is the only it interact with the S2. So here you see this is the how many active amino residue. So here 1, 2, 1 and 2, these 2 amino acid residues, and here also 1. So these 3 amino acid residues are responsible in the RBD part for binding with the S2, and in the S2 there are 2 amino acids like 417 and 453. These 2 amino acid residues are important for binding. So this is all about this the overview. So I'll make some another videos about in detailed information about this kind of molecular structure. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and subscribe my channel. And if you have any queries, kindly write in the comment section. Thanks.